Welcome to this explainer. Okay, so let's dive right into this. In our grand map of metabolism, we know that carbohydrates, fats, and proteins aren't just isolated worlds, right? They're all deeply, beautifully connected. And sitting near the very beginning of one of the most absolutely crucial roads on that entire metabolic map is glucose. So today, we're going to zoom way in. We're going to take one single molecule of glucose, follow it right inside the cell, and figure out exactly what happens to it. But listen, here is my promise to you. I don't want you just mindlessly memorizing 10 random chemical reactions. We are going to treat glycolysis like a fascinating story. Really, think of it as a cellular business venture. We'll look at what the cell is actually trying to accomplish, why it has to spend upfront energy first, where it gets that energy back, and which key steps really control the entire pathway. By the time we produce our final product, pyruvate, you'll understand exactly how we got there. So, here's the roadmap for our journey today. First, entering the cell. Second, the investment phase. Third, splitting the molecule. Fourth, the payoff phase. Fifth, the energy scoreboard. And finally, the pyruvate crossroads. Let's get started.
All right, part 1, entering the cell. To set the scene here, imagine you just ate a massive carbohydrate-heavy meal. You know the feeling. Those carbs are digested down, glucose enters your bloodstream, and eventually that glucose desperately needs to get inside your cells so you can actually use it. But right out of the gate, we hit a biological hurdle. Glucose is a big molecule. It cannot simply squeeze freely across the cell membrane on its own. It needs molecular assistance to cross over, and that assistance comes in the form of GLUT transporters. Now, you'll find that different tissues use entirely different transporters based on their specific real-time needs. GLUT1, for example, is absolutely crucial for your red blood cells in your brain. GLUT3 is the major player for your neurons. GLUT2 handles the heavy lifting in places like the liver and the kidneys, but I really want to draw your attention specifically to GLUT4, which is found in your skeletal muscle and adipose tissue. This is the one you really want to connect back to insulin. Remember that well-fed state we just talked about? After you eat, insulin rises in your bloodstream. In your skeletal muscle and fat tissue, that insulin literally signals the cell to physically increase the number of GLUT4 transporters available at the membrane. More transporters equals more glucose entering the cell. So we're already connecting this directly to the bigger metabolic picture. Fed state, high insulin, abundant glucose, and actively encouraging the body to utilize that glucose.
Moving right along to part 2, the investment phase. Now that glucose has successfully made it inside the cell, welcome to glycolysis. Before we dig into the chemistry, let's establish one absolute, non-negotiable rule. This entire process happens in the cytosol. We have not entered the mitochondria yet. Our basic mission here is pretty simple. We start with a 6-carbon glucose molecule, break it down, and finish with 2 3-carbon pyruvate molecules, capturing usable energy along the way. But, as I mentioned earlier, this operates exactly like a business. Before the cell can make an energy profit, it has to spend upfront capital. So glucose enters the cell, and the very first thing we do is add a phosphate group to it, turning it into glucose 6-phosphate. The enzyme responsible for this in most tissues is hexokinase, and this exact step costs us 1 ATP. The ATP donates a phosphate, becoming ADP, with magnesium actually stepping in to help stabilize those negatively charged phosphate groups. But you might be wondering, why on earth are we spending precious energy right out of the gate just to attach a phosphate? Well, because it does something incredibly useful. It acts like a molecular ankle monitor. You see, glucose could potentially float right back out of the transporter it just came through. But glucose 6-phosphate? No way. It cannot. We have irreversibly trapped it inside the cell. It's staying here, and we're going to use it. That irreversibility is absolutely vital, making this the first of 3 major regulatory steps in glycolysis. Once it's trapped, reaction 2 is just a simple rearrangement. Glucose 6-phosphate is reversibly rearranged into fructose 6-phosphate by the enzyme phosphoglucose isomerase. No carbons are added and no carbons are removed. Before we jump to the next reaction, we have to talk about the enzymes handling that very first trapping step. In most tissues, like we just noted, it's hexokinase. But in the liver and pancreas, we encounter its counterpart, glucokinase. And this is so cool because it brilliantly illustrates how enzyme behavior perfectly matches the body's metabolic situation. Think about how these two compare. Hexokinase has a really high affinity for glucose, meaning it works great even when glucose levels in the blood are relatively low. Glucokinase, on the other hand, has a low affinity but a massive capacity. When you eat a huge meal and an absolute tsunami of glucose comes pouring into your liver via the portal blood, hexokinase would get completely overwhelmed. But glucokinase, it just kicks into gear easily handling that massive post-meal glucose surge. The biology perfectly fits the purpose.
Okay, so we have our fructose 6-phosphate, and now we reach arguably the most important reaction in the entire pathway. Fructose 6-phosphate receives another phosphate group to become fructose 1,6-bisphosphate. This is done by the enzyme phosphofructokinase 1, or PFK1. And yes, we just spent our second ATP. Our cellular business is now down 2 ATP in capital, but PFK1 is a massive deal. It's the major rate-limiting traffic control gate of glycolysis. If PFK1 speeds up, the whole pathway flows faster. If it slows down, glycolysis basically grinds to a halt. So the crucial point here is how this gate is regulated. Think about it. If your cell already has a ton of ATP, does it make sense to burn even more glucose? No, absolutely not. So high levels of ATP and high levels of citrate actually inhibit PFK1. They essentially tell the pathway, hey, we're good on energy, slow down. Conversely, if ATP is actively being burned, AMP levels start to rise. That AMP acts as an alarm bell, activating PFK1 to speed up energy production. Finally, insulin in the well-fed state drives the creation of a very specific molecule called fructose 2,6-bisphosphate, which acts as a massive green light for PFK1. High insulin, lots of food, open the gates and run glycolysis.
Which brings us to part 3, splitting the molecule. We have officially completed the investment phase. We've successfully produced fructose 1,6-bisphosphate, our count is temporarily down to ATP, and the traffic gate is wide open. Now it is time for the physical breaking of our 6-carbon structure. The enzyme aldolase steps up and takes our 6-carbon fructose 1,6-bisphosphate and just cleaves it right down the middle into 2 different 3-carbon molecules, dihydroxyacetone phosphate, which we just call DHAP, and glyceraldehyde 3-phosphate, known as G3P. But there is a slight catch here. The rest of the glycolysis pathway only knows how to work with G3P. It literally cannot use DHAP. So the enzyme triose phosphate isomerase steps in and cleanly converts that DHAP into a second molecule of G3P.
Now, I want you to literally burn this number into your mind right now. 2. From this exact moment forward in the pathway, every single reaction happens twice for every original glucose molecule. We started with 1 glucose, we now have 2 identical molecules of G3P. Keep this mathematical shift in the back of your mind because it is the absolute secret to understanding how we actually end up making an energy profit.
And that brings us beautifully to part 4, the payoff phase. Welcome to the second half of our biochemical pathway. The business has spent its capital, organized its assets into 2 G3P molecules, and now it's finally time to recoup our investments. Time for the payoff. First, our G3P is converted into 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. This is a massive step because NAD+ enters the story. It swoops in, captures high-energy electrons, and becomes NADH. And remember, because we have 2 G3P molecules, this happens twice, netting us 2 NADH. Hold on to those. They are essentially metabolic checks we can cash later for massive energy once we reach the mitochondria. Next up, 1,3-bisphosphoglycerate transfers a phosphate directly to ADP, creating our very first ATP. The enzyme here is phosphoglycerate kinase. This is what we call substrate-level phosphorylation. We aren't using mitochondria here. We are making ATP directly on the glycolytic assembly line. And again, because this happens twice, we just generated 2 ATP. Think about our business math for a second here. We spent 2 ATP earlier, and we just made 2 ATP here. We have perfectly broken even. Everything from here on out is pure profit. Let's trace the remaining steps to get that final profit. 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. Then, 2-phosphoglycerate becomes phosphoenol pyruvate, or PEP, thanks to the enzyme enolase. Now, PEP is a remarkably high-energy molecule. It is perfectly primed for our grand finale. PEP transfers its phosphate to ADP, generating ATP one last time. The enzyme finalizing this whole thing is pyruvate kinase, and our molecule is finally transformed into pyruvate. Since everything happens twice, bingo, we just generated 2 more ADP. But before we go tallying the final score, we absolutely must pause on that final enzyme, pyruvate kinase. This is the third and final major irreversible regulatory step of glycolysis. The control here is just fascinating. Earlier in the pathway, fructose 1,6-bisphosphate actually reaches ahead and activates pyruvate kinase. This is called feed-forward activation. It basically tells the end of the line, hey, we've got material coming down the pipe, keep the traffic moving.
But on the flip side, what happens in the liver during fasting? The hormone glucagon brilliantly puts the brakes on pyruvate kinase. Why would it do that? Well, because during fasting, your liver is desperately trying to make glucose via gluconeogenesis to keep your brain alive. It would be a foolish, entirely futile cycle to aggressively burn glucose through glycolysis while simultaneously trying to manufacture it, right? So glucagon simply shuts this final gate down. It's literally metabolic flux in perfect harmony.
Part 5. The energy scoreboard. All right, the reactions are officially done. Let's pause our pathway progression, step back from the chemistry for just a second, and consolidate the metabolic math. Let's count the money from our cellular business venture. On the input side, we started with exactly 1 molecule of glucose, and during the investment phase, we spent 2 ATP. On the output side, during the payoff phase, we generated 4 ATP, we captured 2 high-energy NADH molecules, and we ended up with 2 molecules of pyruvate. Simple math. 4 ATP generated minus the 2 we spent gives us a beautiful net gain of 2 ATP and 2 NADH for every single molecule of glucose. Our business venture is highly profitable. Part 6. The pyruvate crossroads. So we've got our ATP profit in hand, and we have our newly minted pyruvate molecules. But the story doesn't just abruptly end here. We need to reconnect glycolysis back to the master metabolic map by looking at the branching fates that pyruvate now faces. It is standing at a massive metabolic crossroads, and it basically has 2 main options. If the cell has oxygen and it has healthy mitochondria, which we call the aerobic fate, pyruvate packs its bags and moves right into the mitochondria. There, it gets converted into acetyl-CoA to enter the mighty TCA cycle, while those NADH molecules we made earlier drop off their electrons to drive massive ATP production. But what if the cell doesn't have oxygen? Or what if, like your red blood cells, it simply doesn't have mitochondria at all? Then pyruvate takes the anaerobic fate. It gets converted into lactate. Now let's dispel a huge myth right here, right now. The cell isn't converting pyruvate to lactate just because it's obsessed with making lactate or because lactate is just some useless toxic waste product. No, converting pyruvate to lactate physically forces NADH to drop off its electrons, turning back into NAD+. Remember earlier when NAD+ captured electrons during the payoff phase? If a cell without mitochondria runs out of NAD+, glycolysis completely grinds to a halt. Energy production just dies. By making lactate, the cell actively regenerates NAD+, so glycolysis can keep looping and producing that net 2 ATP to keep the cell alive. Furthermore, that lactate isn't wasted at all. It eventually travels through your blood back to the liver, where the liver uses it to build brand new glucose. It's a beautifully, flawlessly efficient system. And there you have it. Glycolysis isn't just 10 random, disconnected reactions. It's an elegant, logical story. 1 6-carbon glucose enters the cell and gets trapped. We invest 2 ATP to activate it. We split it right in half. We collect our payoff of 4 ATP and 2 NADH. And we cross the finish line with 2 3-carbon pyruvate molecules, heavily regulated by hexokinase, PFK1, and pyruvate kinase along the way. But as we've just seen, this pathway is really just a feeder for something much, much bigger. When oxygen is present, that pyruvate enters the mitochondria and transforms into acetyl-CoA, which leaves us with the ultimate provocative question: what exactly happens when acetyl-CoA enters the grand master crossroads of the TCA cycle? That is a fascinating story all on its own, and we will absolutely follow pyruvate directly into the mitochondria to uncover it in our next explainer. Keep learning, and I'll see you there.